Good afternoon all. Now, today we will discuss about the monohybrid experiment by Mendel. Monohybrid experiment is frequently asked in 4 months. Fine. It is also known as inheritance of one character. As you know, that inheritance of the one character, it means, you know that Mendel decided or selected the 7 characters, but 7 pairs of contrasting traits or factors or alleles or allelomorphs. So here one character is inherited. Fine. So here, before starting the experiment, actually in a textbook, 4 to 5 pages description is there. In textbook, 4 to 5 pages description is there. We have to write 4 to 5 pages according to us. Yes, it is the 4 mark question. So the major question is that we are confused behind this question, that how much portion is written. So in textbook, the much more information is given there, so you understand it properly, that how much portion we have to write there. So before starting this experiment, you have to write suppose here we select the one character. So for example, we select the tall and dwarf plant. So suppose stem height or length of stem, fine, is decided here as a selection for the character. Now you know that stem height it is of 2 types. Now contrasting traits in that tall plant and dwarf plant. Fine. So tall plant it is represented as a capital T and dwarf plant it is represented as a small t. So as you know that in the tall plant and dwarf plant, capital T is here dominant. So here we say that dominant character is there. It is fixed. And small t is recessive. So in previous lecture I have told to you that genes are always in pair and chromosomes are always in pair. So previously before starting the Mendel experiment, I have told to you that these allelomorphs or alleles or factors are nothing but these all are genes. So consider that here capital T and small t. Before understanding this in detail, I want to tell you the new 2 words for today's lecture: genotype and phenotype. Fine. For understanding purpose, genotype Genotype means genetic makeup. Fine. So what you consider here? Genetic makeup means capital T, capital T, capital T, small t, small t, small t. That is genotype. For example, I am telling to you here because understanding easy. Genotype is genetic makeup, so all the alphabets are genotype. Whereas, fine. Then phenotype word. Later on I will tell you that it is characteristic makeup. Phenotype means character, which is expression. So here, for example, tall plant and dwarf plant. These all are phenotype. So you remember, because here the question will be asked in such a way that what is the genotypic ratio, what is phenotypic ratio, like that question will be asked. Fine. So you consider this new two words for you. So stem height, for example, tall plant, it is represented as a capital T, that is dominant. Dwarf plant, that is small t, that is recessive. So previously, as I told to you just before 2 to 3 minutes, that genes are always in pair. So now we select the P generation. So in P generation, we decide the two plants. So here we select the two plants, capital T, and small t. Fine. This capital T plant is selected as a male plant. So this is the symbol of the male plant. Fine. See, this is capital T, so this is genotype. And here capital T, here homologous or homozygous condition is there. Homozygous condition is there, and here it represents tallness. So tall plant is there. So here capital T, that is genotype, and tall plant, it is phenotype. So we say that homozygous dominant tall plant crossed with homozygous recessive dwarf plant. So small t, that is genotype, and dwarf plant, it is phenotype, and we can select it as a female here. Fine. So here this is the P generation, this is male plant, capital T, and this is the female plant, small t. And again this is genotype and this is phenotype. Now you know that organism is haploid or diploid, but it produces gametes by meiosis division. So now here what happened? The new word for you. Third new word, segregation. Segregation here, it is nothing but known as separation. Fine. So now here what happened? At the time of the gamete formation. So here we select the tall plant and dwarf plant as a parental plant. Now here what happened? That gametes are synthesized. So here segregation occurs. So this capital T and capital T both are joined together, and now segregation occurs, means homologous chromosomal pair. So two chromosomes are joined together, they are segregated, means separated from each other, So this produce the two gametes, capital T and capital T. So here in tall plant, two gametes are formed. So number of gametes are 2, but type is only 1. Right? And similarly here, small t, dwarf plant produce the same two gametes, but instead of that capital T here, small t is there. So here we say that it is gametes. And now here meiosis occurs. So tall plant produce the capital T gamete by meiosis division, and small t plant produce the gametes by same meiosis division. So think about here. In small t plant, that is dwarf plant. So what Mendel was done here, Mendel is in dwarf plant, first of all emasculation occurs. So with the help of that, yes, previous lecture I have told to you that Pisum centarum flower is bisexual flower. So Mendel had select the dwarf plant, select the bud of the pea flower, fine, and with the help of the two forceps, two anthers were removed. Then bagging can be performed, and that's why here in the dwarf plant I put the female symbol here. Now after ripening of the fruit, ripening of the flower, so remove the bagging which was done on the dwarf plant, then select the tall plant pollen grains and spread over it. Fine. So here what happened? So these are pollen grains and these are ovum. So now carefully understand it. Capital T with small t. So first gamete of the male plant and first gamete of the female plant. So here capital T and small t both are united together and form the capital T small t. So this is zygote. Or this is F1 generation. So gametes are fused and in F1 generation progeny are formed. So these one are zygote. So genotypically capital T and small t same first and second. So here what I can write, capital T small t again. Same way, second gamete and first gamete. So what I can write, capital T small t, and similarly this one. So this all are F1 generation zygotes. So by observation you see that here 4 number of offsprings are formed. 4 offsprings are formed and all are tall. So here tall plant, here same way tall plant, here same way tall plant, and here same way tall plant. So here what happened? In the F1 generation, 4 offsprings are formed, and out of that 4 means 100% plants are heterozygous tall. So now here what you understand, that in P generation, homozygous tall plant cross with homozygous dwarf plant, or we say that homozygous dominant plant cross with homozygous recessive plant. So in F1 generation, all are heterozygous tall. So here in heterozygous tall condition, fine, heterozygous tall condition, capital T is along with that small t, but remember that small t is unable to represent its character. That's why we say like this, that small t is recessive, because capital T is along with the small t, but expression of the character that is of the capital T only. So we say that heterozygous tall plant here, or you use that word homozygous dominant cross with homozygous recessive. As a result, in F1 generation, all the plants are heterozygous dominant. Okay, now here what we do in F1 generation, if now what we use that word self fertilization here, that here we have to select the same plant, one plant. Select the two plants from here in the F1 generation. Select the one plant as a male and one plant as a female, randomly, no problem. So capital T small t cross with capital T small t. So here you select the one plant as a male, and here you know that phenotype is tall plant, and here same way phenotype is tall plant, and we select this plant as a female. Now according to this, you form the gametes here. So this is the F1 generation, two plants, one plant as a male and one plant as a female. So now here two gametes are formed. As you know that number of gametes are 2. So here capital T is with the small t. So segregation occurs. So in one gamete capital T is there, in another gamete small t is there. So here number of gametes are 2 and types are also 2. Whereas here number of gametes are 2 but type is only 1. Similarly here two different types of the gametes are formed. One is capital T and one is small t. So now we can write the F2 generation zygotes. So in F2 generation what happened? Fine. See, one by one we will cross it. So capital T now cross with this capital T. So here what happened? Capital T is there. Capital T, capital T. Same way, capital T is now met with this small t. So you write capital T and small t here. Now small t cross with capital T. So please remember that majority we write the capital letters first. So capital T and small t here we can write in this way. And same way the last gamete, that is small t and small t fuse together and form the zygote, that is small t, small t. So as you now understand that these all are genotype of the zygotes. So now here capital T, capital T. This genotype represents which phenotype? Tall plant. This represents tall plant. This represents tall plant. And and this represents dwarf plant. So here in F2 generation, number of offsprings are 4. So out of the 4 offsprings means 3. Out of the 4 offsprings, that is 3. So here 75% proportion is of tall plant. And out of these 4, only 1 consists. That is 25% dwarf plant. So basically you understand this experiment. Fine. So here I can stop now. Monohybrid experiment remaining 10 we will discuss in the next lecture.